Hi everyone, it's my pleasure to welcome you to Inside Quantoom, episode 2. In this episode, I'll share Quantoom's vision and tell you how we're bringing radical innovation to the entire mRNA workflow, from discovery all the way to development and large-scale production of mRNA. This episode is very special as we are officially launching our revolutionary Ntensify technology for mRNA production, which is now commercially available. I leave the floor to Guillaume, our head of product management and marketing, and Lionel, our product manager, who will present our technology production called Ntensify, as well as its incredible benefits. To produce RNA, a good DNA template is needed, and we also develop a disruptive process for that production. Irina, our head of R&D, will give you a sneak preview of our cell-free linear DNA production services. Then Regis, our VP of Global Sales, will wrap up the session with a few take-home messages. Eventually, before we close, the team will answer your questions in a live Q&A session. However, the chat is already open, so feel free to send us any questions that you might have during the presentations, and we will make sure that we answer them during the Q&A session. Now, before jumping into our RNA and DNA production technologies, let me remind you who we are and where we come from. 10 years ago, we founded Univercells, a company with one mission in mind: to make biologics available to all. Many people and countries can't afford the medicines they need, and we strongly believe in technology-driven affordability. The technologies we develop at Univercells are designed to lower the barriers to low-footprint distributed manufacturing, a way to make biologics regionally at affordable cost while still being capable of producing large quantities. The COVID-19 pandemic has been a wake-up call for all of us. If still necessary, it evidenced the urgent need for innovative technologies, intensified and flexible enough to support rapid responses and vaccines on demand in case of pandemics. We indeed experienced that the countries that were not equipped with up-and-running biomanufacturing infrastructure did not receive doses enough and doses on time, showing again that the current centralized manufacturing model is unable to support rapidly and affordably the whole world, and that distribution actually of production instead of distribution of the product might be the most efficient way to provide complex drugs in an equal manner. This is why in June 2021, we decided to launch Quantoom Biosciences with the goal to revolutionize the way mRNA is produced. Looking at mRNA vaccines and actually other drugs, we think there are still several barriers to overcome. From the identification of human pathogen to the commercial production of an efficacious mRNA vaccine, we identified several complex steps. It is complex to design the optimal RNA sequence, identify and down select the best mRNA candidates. It is complex to develop the right production process with the corresponding necessary reagents. It is complex to scale it up and validate it for clinical studies and for commercial production. Now let me explain the way we develop our production solution. At Univercells, we always start with a radical redesign of the process, having in mind the goals I'll describe right now. So we can illustrate our approach with a pyramid that actually you should see on the screen, you should see it. And as you can see on top of it, there is the process. It was designed to provide all the benefits of continuous production while using very little batches of only 20 milliliters operated in sequential mode. It was designed to avoid scaling up and its related risks. It was optimized to end up with only one purification step, and it was optimized to be mRNA construct agnostic, delivering one single process for any RNA construct to be produced. All the know-how we developed around this mRNA construct agnostic process is actually delivered through mixes of reagents. That's the second layer of the pyramid you can see. Those mixes contain all the reagents at the right concentrations, volumes, ratios, in the right buffers to guarantee process performance. These mixes of reagents are used in single-use disposable kits that have been optimally designed. And finally, the fourth layer at the bottom of the pyramid, we have the equipment, so the system that was developed around the disposables for the implementation of the redesigned optimal process. This results in a low footprint, integrated, automated system that operates the entire manufacturing process. And all this provides a de-risked method for the small to large-scale production of mRNA, providing consistent results for any mRNA construct. Now, before leaving the floor to my colleagues, let me mention that the mRNA technology we launched today is part of a complete platform called Infinity, made of 3 pillars. Amplify, the first pillar, is designed for the synthetic production of linear DNA. Ntensify, the second pillar, is the main focus of our webinar today. And Encapsulate, the third pillar, is designed for the production and purification of lipid nanoparticles encapsulated in RNA. I will now leave the floor to my colleagues who will tell you more about the benefits of the technology we developed, and we'll see you afterwards for the Q&A session. Thank you so much for your attention.
Thank you, José. Hi, I'm Guillaume, Head of Product Management and Marketing at Quantoom Biosciences. Today, I'm proud to represent our team of scientists and engineers who have dedicated countless hours to bringing radical innovation to the way mRNA is produced. Today, we are announcing the launch of Ntensify, a line of production technologies that embody the pinnacle of our efforts. Essentially, Ntensify are low-footprint, automated production technologies able to synthesize and purify short to very long messenger RNAs in bulk, using Quantoom's reagent premixes to maximize yield, quality, and cost-effectiveness, starting from a chosen DNA template. This produces purified naked RNA from the Ntensify, ready to be formulated into a drug product. But Ntensify is more than just equipment. Following the very same principles previously explained by José, Ntensify has been specifically designed for all developers of mRNA vaccines and therapeutics along their journey from early candidate discovery up to commercial production. Our design brings 3 main benefits to mRNA programs. 1. De-risk process. The Ntensify technology was originally based on a process transferred from our partner Eterna, which we have jointly optimized over hundreds of upstream and downstream reactions with 18 constructs from 1,000 nucleotides to 11,000 nucleotides in length. Today, we dare to say that Ntensify is built on an optimal process, delivering an above standard performance, both in yield and quality, fit for various types of RNA constructs. 2. Speed to market. Ntensify accelerates product development in 2 ways. The first is during the discovery phase with high-throughput candidate screening, and the second is the ability to produce microgram up to multi-kilogram RNA drug substance without spending several months scaling up beyond 20 milliliter. 3. Cost efficiency. By using our Ntensify production technologies, developers can benefit from massive savings along their journey. Our intensification and chaining principles gives Ntensify a low footprint and the ability to handle smaller volumes than conventional approaches, impacting the required size of the production facility and the related capital expenditures. That said, most savings will be operational and linked to reagents, thanks to our optimized process with high titers and recoveries, but also lower use of reagents for equivalent output, but also time and materials savings thanks to our process design, which virtually eliminates scale-up, and rather keep the process at a maximum scale of 20 milliliters, and then iterating it in multiple times. And finally, the automation and integration of Ntensify that simplifies the production workflow, allowing for lower batch failures and saving on material losses. Stay online, we will illustrate those benefits with data later in the presentation. Ntensify comes as a suite of 3 distinct models to match the needs of RNA developers at every stage of their journey. We have the Mini, the MIDI, and the Maxi. The Mini version is a powerful solution offered to research and development scientists to screen many different constructs in search of the ideal RNA candidate. but it can also produce milligram quantities of a particular RNA construct, an ideal companion for in vivo studies. The MIDI version is the centerpiece within our Ntensify production technology line. Our Ntensify MIDI technology is compliant with good manufacturing practices and allows developers to produce 1 to 5 grams of RNA per day in a single batch, the equivalent of more than 7 million doses on an annual basis, adequate for all clinical trial stage and mid-capacity commercial production. Finally, our Ntensify MIDI will soon be available for an upgrade to the Maxi version. This will allow developers to drastically increase their batch size by running the system continuously over a period of 6 days. Our Maxi version is the ideal tool for developers looking for kilogram production capacities, the equivalent of more than 50 million doses of a prophylactic vaccine on an annual basis. In a nutshell, that was an introduction to Ntensify. Let's now deep dive into the specifics of the Ntensify Mini and MIDI models available today. Lionel, over to you.
Hello everyone, my name is Lionel. I'm product manager at Quantoom Biosciences, and this is my pleasure to spend time with you today to talk about our Ntensify Mini. The Ntensify Mini, the first to come in our Ntensify production technology line, is an RNA production system at small scale intended for research and development purpose. This is typically an R&D screening system using multi-well plates with 100 microliter in each well, operating our redesigned process to provide hundreds of micrograms of RNA. This system is suitable for screening purpose with 2 applications in mind. The first one is for RNA discovery application. The system can accommodate up to 192 different DNA templates, with 1 DNA template per well to produce around 500 micrograms of purified RNA for each construct. The second application is for preclinical development. A single DNA template can be transcribed in all the 192 wells to produce over 100 mg of particular purified RNA. In terms of dimension, our Ntensify Mini is 2.5 meter wide, 2.3 meter height, and 1 meter in depth, resulting in a footprint of only 2 square meters. In operation, the system weighs around 1 ton. The utilities that are required are electricity and wired access to internet, and the system can be operated in a standard laboratory environment. Now that you have a good understanding of how the Ntensify Mini is designed, I propose that we have a closer look at how the system operates. As you can see, our Ntensify Mini is made up of 1 benchtop instrument under a unidirectional flow. Before starting your production, you will load here tips, the reagent premixes, and multi-well plates containing your DNA templates. All the reagents that are required to synthesize purified mRNA are available as a ready-to-use premixes for 20 ml of in vitro reaction. They meet the highest quality standards and are manufactured to enable optimal process performance in yield and quality within the Ntensify Mini. In terms of single-use components, all these are provided by Quantoom and selected to be used in the Ntensify Mini in a R&D context. Once started, all mixing and transfer operation by the robot will take place at the working station here. The RNA synthesis station located here houses up to 2 multi-well plates containing 96 wells of 100 microliter each, where the in vitro transcription occurs. After the IVT, the reaction media is pretreated and transferred in deep well plates for purification. The purification process using silica-coated magnetic beads begins at the station here, where RNA will be captured and extensively purified. Finally, the purified RNA is harvested either by the operator in the purification plates if several different constructs have been produced, or it can be harvested by the robot in 50 ml tubes as a pool for single construct production. As you can see, our Ntensify Mini was designed to address our customers' specific needs. On one hand, using our system for your RNA discovery application, we allow you to produce small quantities of an incredibly large number of constructs in a very short time with high quality and reproducibility. On the other hand, using our system for your preclinical development, we allow you to do multiple preclinical studies with the same batch of RNA, with a direct transfer from screening to preclinic of your best candidates, and all of this at the same RNA quality level as your future clinical studies, using exactly the same intensified and optimal process that will be used at larger scale. All these features ultimately de-risk the production of multiple candidates or small batches of particular RNA, as our predefined process guarantees the output in terms of yield and product quality. You can focus on your research, giving you more opportunities to identify the right candidates, eliminating variability for more accurate results, and accelerating your preclinical phase to fast-track vaccine and therapeutics development. All in all, we hope you have all the information you need to appreciate that our Ntensify Mini is the perfect bridge from discovery phases to clinical studies. I will now give the floor back to my colleague Guillaume, who will tell you more about our Ntensify MIDI system.
Let's now dive into our flagship Ntensify MIDI. The Ntensify MIDI is a centerpiece in the product line. It's an automated RNA production system running on the same redesigned and optimized process as the Ntensify Mini, but at 20 ml scale, with a capacity of 1 to 5 grams of purified naked RNA per batch. Ideal for clinical trials and mid-capacity commercial production, able to produce the equivalent of up to 7 million mRNA doses per year. Looking at the dimensions of our Ntensify Mini, the system is 2 meters wide, 2.4 meters high, and just 1 meter in depth, which gives it a footprint of only 2 square meters. The system weighs approximately 1 metric ton. The utilities that are required are electricity and wired access to the internet. The system is designed to be operated in an ISO 8, so Class D, or ISO 7, Class C environment. I'm now standing next to the system and would like to guide you through how our system operates. Here is our Ntensify MIDI, which is made up of a closed process chamber with 2 automated robotic arms under a unidirectional flow. Zooming in on the system's plan deck, we can divide it into 2 parts. The left part is where the RNA synthesis will occur, while the right one is dedicated to the purification of the crude RNA. Before starting the production, the first step is to position the reactors and the purification vessels. Place the reagent premixes on their dedicated areas and connect them to their specific assemblies. All those are provided in the form of ready-to-use kits for a batch. The RNA synthesis station houses 1 to 4 reactors. Each reactor contains wells of 20 milliliters for a total of 200 milliliters of in vitro transcription. The 11th well is for the sensor that continuously monitors and controls the temperature during the process. Once the production has started, reagents will be mixed and heated up in the premix tank, and dispensed in a sequential continuous manner by the robotic arm within the 20 milliliters wells to initiate the RNA synthesis. After the in vitro transcription, the reaction media is pre-treated in each well and then pulled together at the pooling station. Now, the purification process takes place within specifically designed vessels at the far right of the process chamber. It uses silica-coated magnetic beads captured by the magnetic stent to harvest the purified RNA. Finally, the purified drug substance is continuously collected by the robotic arm in bulk. Now that you know how it all works, let us illustrate the benefits of our Ntensify approach by assuming that you're an mRNA drug developer interested in producing 50 million doses of mRNA vaccines, with 42 batches per year, comparing Ntensify against a so-called conventional approach at 40-liter scale. For this purpose, we will demonstrate the value Ntensify could generate once upgraded to its bigger capacity version, the Ntensify Maxi. The figures for each approach and related savings with Ntensify are based on our cost modeling tools. The 3 main benefits of Ntensify mentioned earlier, de-risk process, speed to market, and cost efficiency, can typically be achieved through the following 4 levers. 1. Less reagents for more high-quality drug substance. 2. No scale-up needed. 3. Intensified and chained workflow. 4. Low footprint facility. Reagents are the largest cost driver for mRNA manufacturing. Therefore, we worked on the RNA IVT and purification process to do more with less, while still providing a superior quality drug substance. Our Ntensify Maxi, thanks to our optimal process, handles volume of only 13 liters of IVT per batch, compared to 40 liters in a conventional approach. This is threefold decrease in the volumes to be managed. Our process also consumes less reagents per unit volume of product, such as capping reagents or enzyme cocktails, and less purification and filtration media, as there is only one purification step, for the same output and a quality above specification. Now, assuming that the 40 liters is achieved with a conventional approach, scaling up the production by a factor of 20,000 from the laboratory microliter scale is no easy task. It typically follows a staircase approach that takes time and resources, 18 months and 8 million euros in materials and FTE cost. Ntensify stays at 20 ml scale and can be operated in continuous batch mode without the need for scale-up to produce the quantities required, saving 15 months and about 7.5 million euros. As said previously, Ntensify integrates both the 20 ml IVT and one single-step purification in an automated system. That significantly lowers the batch failure rate in routine operations. If you consider the volumes and the cost of reagents for each batch, Ntensify allows savings of more than 6 million euros in annual operating cost. Finally, assuming an approach that uses container-based clean rooms, Ntensify fits into one 30 square meter container. And with its ready-to-use reagent premixes, it does not require media and buffer preparation room. On top of that, the lower values involved require less warehouse spaces compared to a conventional approach. All in all, Ntensify makes the annual production of 50 million doses of an mRNA vaccine possible in a fully equipped facility of 300 square meters instead of 800 square meters, generating an additional 4 million euro in capex savings. We hope that this illustration of the benefits of Ntensify with concrete figures was useful to you. Our teams are at your disposal should you be interested in knowing more. Before closing the chapter on the Ntensify production technologies, you might be aware that the critical, if not the most critical input, is the DNA template for the production of RNA. My colleague, Irina, will introduce you to our DNA service to make sure that our Ntensify customers can rapidly source the right DNA templates at the right quality.
Thank you, Guillaume. Hello everyone, I'm Irina Ibalou, Head of Research at Quantoom Bioscience. I am delighted to introduce our DNA on-demand service from our facilities in France. Our service starts with plasmid assembly and continues up to cell-free linear DNA production, using our Ntensify technology to guarantee the secure source of our high-quality grade DNA templates, suitable for our Ntensify system for drug discovery application or preclinical and clinical development. We offer a range of services to support your mRNA development, such as plasmid DNA engineering. So from your in silico sequence, we can build the vector containing the poly A-tailed gene of interest and provide you with the purified template and the certificate of analysis. We can build a research cell bank for each plasmid. And once the template is available, our Amplify experts will screen for the amplification parameters. Depending on your downstream application, we can provide high-throughput production of cell-free linear DNA constructs at small scale, or larger batches of linear DNA up to 70 mg. Each batch is delivered with a robust analytical package that includes NGS, DNA integrity and purity, or endotoxin assay. For our Ntensify users who require rapid access to a safe and clean DNA supply as a starting material for RNA production, our DNA on-demand service offers cell-free linear DNA production in less than 6 weeks. This means that you will be able to secure your DNA source, accelerate your preclinical and commercial product development, and de-risk residual contamination of your end product. Therefore, unlike DNA manufacturing competitors, we deliver the most suitable DNA for your mRNA synthesis with no additional linearization steps and ready to use in your Ntensify system. Thank you for your attention. I'm now letting Regis take the opportunity to conclude in our studio.
I'd like to thank all my colleagues who have provided you with information about who we are and what we do. I'm Régis Leonard, Vice President of Sales at Quantoom Bioscience. It's time to wrap up this webinar, and I'd like to make it simple. If you're a biopharma company already developing vaccines but eager to leapfrog your competitors, if you are a biotechnology company eager to issue your first promising vaccine candidate and be the first one to market it, if you're a CDMO helping companies to develop and mass produce their new vaccine, eager to differentiate yourself from the competition, and you work in human or animal health, what's probably keep you up at night is how to overcome the barriers José already shared and develop the much needed vaccine that the world is eagerly awaiting and that would bring significant benefits to all. So you are at the right place and the right time with us today. Indeed, we are pleased to announce that our Ntensify Mini and Ntensify Midi are available for you. These systems were created with a singular purpose in mind: to simplify your daily life. Why is that? One, we are the unique company able to supply the equipment, the plastic disposables, the reagents, and related DNA services, eliminating the burden to coordinate with multiple suppliers. Two, we offer a one-kind, fully automated process for manufacturing mRNA, capable of producing from micrograms to kilograms per year in a single low-footprint instrument. Three, our underlying process is ready for use, fully de-risked, and offers significant savings with a threefold decrease in volume reagent. So, we believe that Quantoom Bioscience has definitively created a paradigm shift in mRNA vaccine production. The way to produce vaccines will be different from now on compared to what it went before. The future looks brighter, and the future starts now. So if you also want to experience all the mentioned benefits like our first customers will, and leapfrog your competitors in your industry, connect with us at bd@quantoom.com. We would be very happy to demonstrate the system to you. Thank you, and talk to you soon.